Welcome to today's Explainer, where we're looking at a breakthrough so massive, it's literally going to change how we understand the brain. And the crazy thing? It all starts with an organ physically the size of a single grain of sand. We're talking about the creation of a fully three-dimensional map of an actual brain. This isn't just about showing us where neurons live. It actually reveals the intricate, real-time connections and interactions of these neurons during complex behaviors. So we're taking a journey today, from a tiny little bug to the absolute future of human neuroscience, and trust me, it's going to blow your mind.
Now, this entire breakthrough revolves around something called Connectomics, is a rapidly developing sub-branch of neuroscience that maps neural connections to figure out exactly how they drive different types of behavior. For the longest time, we've realized that when it comes to the brain, nothing is truly localized to just one single isolated point. Any action, any thought you have relies on these incredibly complex networks spread all over the brain. Different thoughts or actions activate completely different networks, or as they're called, connectomes. So mapping out these networks is absolutely the ultimate key to understanding how a brain actually functions.
Moving into Section 1, Why the Fruit Fly? To pull off mapping a complete connectome, researchers needed a brain they already understood inside and out. Enter the fruit fly. Now, I know you might just see them as that really annoying kitchen pest, but to biological researchers, they're absolute treasure troves. They've got very simple genetics, they're super easy to grow in a lab, and they have this really rapid life cycle. But here's the real kicker. Despite having a brain the size of a literal grain of sand, these flies exhibit highly complex behaviors. They're sexually dimorphic, meaning males and females differ, and they actually have these intricate courtship rituals. We're talking about flies doing specific dances or even singing to attract mates. That means there's just a vast, incredible amount of complexity packed into a remarkably tiny space.
And here's why this matters to us. 60% of the genes inside these fruit flies are actually pretty similar to human genes. Even more staggering, up to 75% of non-human disease genes have an actual match in these flies. So, because they're so easy to manipulate genetically, and because we honestly share so much biological foundation with them, they're just the absolute perfect stepping stone for human medicine and ultimately mapping the architecture of the mind.
Which brings us to Section 2, 21 million brain slices.
Let's look at the history here. Back in 1986, a team of researchers released a study where they mapped the 302 neurons of a simple roundworm called C. elegans. But get this, it took them about 20 years to complete. Why? Because they were literally using colored pencils to trace the neurons manually by hand. Just think about that. Fast forward to the early 2000s, and the digital computing boom finally gave us the power to modernize this process. By 2021, with vast resources pouring in, Google mapped the very first sections of the fruit fly brain. And today, the full, complete map has been officially published in the journal Nature.
The leap in technology here, it really can't be overstated. Going from colored pencils to artificial intelligence literally changed the game. The researchers partnered up with Google and utilized some cutting-edge AI developed by Princeton University. Honestly, without artificial intelligence jumping in to quickly identify individual neurons and trace all these insane connections, compiling this modern map would have taken an impossibly long time. AI is what allowed scientists to trace the entire brain network and discover every single connection super fast.
But let's take a second to really comprehend the scale of the physical work that had to happen before the AI even stepped in. To map this microscopic brain, researchers had to physically slice it up. They cut this single sand grain-sized brain into 7,000 individual, unimaginably tiny layers.
And what's truly staggering is the sheer volume of visual data those slices created. Those 7,000 tiny layers, they were scanned by computers to produce a monumental 21 million individual images. 21 million images that all had to be compiled, meticulously stitched together, and analyzed to form one single, cohesive, three-dimensional map. Out of that massive sea of data, an absolute masterpiece emerged. The finished connectome revealed 50 million distinct connections between the fly's 140,000 neurons. Every single connection, every single neuron, all mapped with ridiculous pinpoint precision. We now officially know the entire brain network of an adult fruit fly, identifying every individual function that allows it to sing, dance, and just live its life.
That takes us to Section 3, Simulating Life Inside Computers.
The discoveries from this were immediate. Out of the approximately 8,500 known and named cells in this brain, researchers found that about 500 of them were completely new, previously unknown neurons.
Think about that. We have studied these flies for well over a century, yet this connectome instantly revealed neurons we have literally never seen before in science. It shows us exactly how these neurons interact to produce specific types of behavior.
But here is where it gets genuinely futuristic. Because this entire connectome is essentially living inside a computer now, researchers can actually start manipulating it. Philip Schlegel and his team have already used this map to activate virtual neurons and simulate real-life behaviors. For example, they simulated something called the proboscis extension, which is basically just the fly extending its tiny straw to suck in some sugar. But they didn't use a real fly. They just activated the virtual neurons inside the computer simulation, mimicking everything perfectly. We can now study brain circuits and test complex behaviors entirely virtually.
Let's wrap up with Section 4, the next 100 years.
Now, the physical scale of what lies ahead is frankly staggering. Remember, what we're looking at right now are just baby steps. Back in 1986, it took 2 decades to map the worm's 302 neurons. Today, we've conquered the fly's 140,000 neurons. But look at our next major target, the lab mouse. A mouse has 70 million neurons. That's about 700 times more than the fruit fly. We're still decades away from being able to map that. And the ultimate target? The human brain. We possess 86 billion neurons, forming trillions of connections. If we assume the same rate of technological progress, mapping the human mind might still be up to 100 years away.
But despite the massive timeline ahead of us, this achievement simply cannot be understated. As the researchers pointed out, this is a really important first step that one day might help us understand everything about ourselves. By building the tools, the AI, and the methodologies to map this fruit fly, we've laid down the foundation. This will inevitably lead to major breakthroughs across medicine, computer science, and honestly, almost every scientific field you can imagine.
We've already seen how researchers can trigger virtual neurons to make a digital fruit fly eat. It really makes you pause and think about the horizon of this technology, doesn't it? If we are laying the groundwork today that's eventually going to lead to mapping the 86 billion neurons in our own heads, well, what happens when we can finally simulate ourselves? What profound mysteries of the human condition will we unlock when the mind is fully mapped? It's a fascinating question, and one that science is finally, steadily crawling toward. Thanks for joining me on this explainer, and I hope it sparked your curiosity about the incredible universe inside our own heads.